Hi, my name is Mogan Gim, and I am the author and presenter of this paper titled ArkDTA, Attention Regularization Guided by Non-Covalent Interactions for Explainable Drug Target Binding Affinity Prediction. This paper was accepted by the ISMB conference and has been already published in the Bioinformatics Journal. First and foremost, I would like to express my gratitude towards my lab colleagues and my principal investigator, Professor Jae Kang, for their support and valuable feedback which helped me complete this publication. Here is a list of the authors and their affiliations and their roles as well. First, I will present a simple explanation about our background and also the preliminaries, and then I will go in depth into explaining our model architecture for ArkDTA and its associated attention regularization method as well. Next, I will briefly explain the results of our experiments and also our qualitative analysis on our ArkDTA model. Last but not least, I would like to conclude my presentation with our limitations and future work. So basically, the main task of our paper is to predict binding affinities given protein-ligand pair input using structure-free model-based approaches. There have been lots of structure-free models which presented and proposed innovative attention mechanisms and various deep learning architectures as well. Meanwhile, non-covalent interactions are one of the most important chemical concepts in predicting drug target binding affinities. However, only very few studies using structure-free prediction models address the importance of this concept. Therefore, we set this design goal onto formulating our own approach. Our design goal is to incorporate the important chemical aspects in current attention mechanism and provide better explainability to actual users. In order to achieve this design goal, we set up a solution by devising a novel regularization method that modulates the distribution of cross-model attention weights. Our paper title, ArkDTA, actually stands for Attention Regularization Guided by Non-Covalent Interactions. In this paper, our main emphasis and focus is to regularize the attention mechanism based on the actual chemical-related non-covalent interactions between the protein target and the drug compound. We believe that these interactions would be helpful in not only predicting binding affinities with higher accuracy, but also offering better model explainability as well. First, I will talk about the preliminaries for our model. For our data representation, we have two types of input data, which are the protein data and the ligand data. The initial input protein data is a FASTA code containing a sequence of amino acid letters. To encode this data, we use an evolutionary scale modeling, which is a pre-trained protein language model. This model has a very large space of parameters and has been trained on almost 10 millions of protein sequences. As a result of this encoder model, we get to have a set-based input protein data, which is represented as a set of amino acid residues containing protein structural conformational features. Proteins are long amino acid sequences with arbitrary lengths. Each amino acid residue, regardless of the same letters, has a unique structural characteristics encoded by the ESM model. On the other hand, for the ligand data, we use a SMILE string that describes the chemical structure of a compound. To encode this SMILE string, we use a ArkDTA tool by converting it into a Mogan fingerprint. We use the 1 bits in the Mogan fingerprint to extract a set of chemical substructures. Our rationale is the following. Understanding the molecular substructures of drug compounds is important in drug discovery industry. Identifying which substructures are relevant to protein-ligand interaction is essential in medicinal chemistry. Perhaps the main aspect of our model is to incorporate non-covalent interaction labels. We analyzed the non-covalent protein-ligand interactions of binding complexes contained in the PDB-bind dataset using the protein-ligand interaction profile, abbreviated as a PLIP tool. Initially, we have multiple types of non-covalent interaction labels, but we aggregated them into a binary label indicating the presence of any type of non-covalent interaction between a protein residue and a compound's atom. However, we also did an additional aggregation by making it into residue-wise non-covalent interaction labels. This is the overview for our model architecture for ArkDTA. ArkDTA consists of 4 different modules. A, which is the protein encoder module, B, which is the ligand encoder module, C, which is the protein ligand integration module that consists of 2 sub-modules, ArkMAB and PMA, which stands for multi-attention block based on attention regularization guided by non-covalent interactions, and the pooling by multi-head attention layer. Last but not the least, we have D, the binding affinity prediction module, which we will not explain further in this presentation. Our main intuition for this model architecture is to model the initial states of the individual input data, which are the drug compound, the ligand, and the protein data as well. After using each of its individual encoders, we treat them as a set of embeddings, and we use them to decode into a binding complex embedding like below. In this case, this model relies heavily on the C model, which is the integration model. We will move on to explain each of these submodels and how we incorporated the non-covalent interactions. But first, I will give you a brief explanation about how we managed to design both of these encoder modules. For the protein encoder module, the input is a sequence of amino acids, and the output is the set of amino acid residue embeddings containing the protein structural features. We have 2 different design rationales. First, most DTI datasets contain limited amount of protein data available for training. The ESM model is pre-trained on massive amounts of protein sequences. Therefore, we figured that leveraging its pre-trained parameters may compensate for this limited amount of issue. The second rationale is that regardless of the same amino acid letters in the FASTA sequence, each amino acid residue has a unique contextual and structural characteristics encoded by the ESM. This provides the basis for treating these embeddings as elements of a protein set. For the ligand encoder module, we use the Mogan fingerprint to extract the corresponding chemical substructures of a given molecule. Chemical substructures are one of the essential features in drug design and reproposing strategies, such as functional groups or scaffolds. This also provides another basis for treating these chemical substructures as elements of the ligand set. For the protein ligand integration module, the input is obviously a set of residues and a set of chemical substructures, while the output is a single binding complex embedding. Like previously mentioned before, ArkMAB uses an attention regularization guided by the non-covalent interactions, while the PMA layer aggregates the refined residue embeddings into a chemically refined residue embeddings. The basic idea of attention mechanism is to assign normalized attention weights based on the pairwise scores between the cories, which are the protein residues in this case, and the keys, which are the chemical substructures. Based on the distribution of the attention weights, we can only infer which part of drug compounds are highly relevant to the DTI. But some inactive residues, without NCIs, do not have any chemical interactions with the incoming drug compound. So we raised this question, whether these attention weights actually offer model explainability. We first thought of directly regularizing this whole attention weight distribution. However, attention mechanism is an unsupervised component, and directly regularizing it would be a rational choice. But we thought, what if we append a pseudo-key embedding, known as the pseudochemical substructure, and add it to the current set of actual chemical substructures? And our strategy was to encourage the attention mechanism to distribute almost all the weights to the pseudochemical substructure when the partner residue does not have any actual chemical interaction. Note that all weights should be summed up to 1. So meanwhile, if the pseudochemical substructure has monopolized all of the weights, then the rest of them distributed to the actual chemical substructures will be almost near to 0. But at the same time, we want to make sure that the weights are unsupervisedly distributed to the actual chemical structures and less to the pseudochemical substructure if the residue has an actual NCI. This is how we manage to modulate the attention mechanism without going against the unsupervised manner of their original attention mechanism approach. In this case, by devising this kind of strategy, we get to have two different model explainability points. We can now interpret which residues are identified as having NCIs by the model. And also, we can now interpret which chemical substructures having the NCI with some specific residues are identified as being highly relevant in predicting binding affinities by the model. For example, for methionine, we can see that the attention weights distributed to the actual chemical substructures are summed up to 1, while the pseudochemical structure is almost to 0. We can infer that this particular type of the amino acid residue is regarded as having an NCI with the incoming ligand. But on the other hand, other residues such as the cysteine have no such attention weights distributed to the actual chemical structures, but almost to the pseudochemical substructure that is nearly to 0.999. In this case, we can see that how the model is reasoning based on the chemical knowledge it has been pre-trained before. So in order to help the model modulate the attention weights based on the given chemical knowledge known as non-covalent interactions, we first extract the attention weights from this ArkMAB and then re-aggregate it into NCI prediction scores as if that we are treating it as a binary classification task. We then use the extracted NCI labels that were previously engineered by the PLIEP tool and regard them as ground truths for modulating the attention weights. So the loss subjective between the two different scores and the labels is the cross entropy since it is a binary classification. So based on this loss subjective, we expect the model to formulate a more chemical-like attention weight distribution inside its ArkMAB block. Here is another diagram that describes how the ArkMAB looks like in the inside. And you may notice that the pseudovector is appended to the current set of the chemical substructures. And also we get to employ attention regularization based on this auxiliary loss objective that was described in the previous slide. This is how we managed to help the model distribute its attention weights based on this learned knowledge of non-covalent interactions. As a result, we get to obtain chemically refined residue embeddings, and now we get to use them to aggregate them into a one single-handed binding complex embedding, just like in the next slide. In this case, we get to use two different virtual seed vectors and by means of pooling the arbitrary number of residues based on attention mechanism. In this case, our main intuition is to help the model distinguish the residues with NCIs and the others without NCIs using two different seed vectors. As a result, we have two different embeddings. We concatenate them together and then reduce their dimension into the same dimension of the original embeddings, and that makes the final output of this whole drug target protein ligand integration module, known as the binding complex embedding. So the design rationale, again, is the following two points. ArkMAB is intuitively based on the concept of the conformational changes in target proteins induced by their partner ligands, where the underlying chemical aspect is the non-covalent interactions. PMA, which stands for pooling by multi-ad attention, helps aggregate the set of residues into two distinct groups, supposedly those with NCIs and others without, into one single-handed binding complex embedding. This actually aligns with the ligand being docked into certain residue regions of the target protein. Here are the experimental results on the binding affinity prediction. As you can see the results, our model wasn't able to outperform all types of evaluation metrics, especially in the KIKD subset of the PDB-bind dataset. However, we managed to outperform other baseline models in the IC50 subset, and we concluded that the attention realization method does seem to offer some benefit into improving the accuracy of binding affinity prediction. Here are the qualitative analysis results of the case studies of our attention weights extracted from the ArkDTA. As you can see, the attention map itself is pretty sparse, where the actual weights are distributed to the actual chemical substructures when the corresponding protein residue has an actual NCI. This offers explainability in terms of which protein regions contribute to the binding affinities when being interacting with the incoming ligand. At the same time, we can also look into some certain chemical substructures with high attention scores and examine then which of the parts of the compound do contribute to this binding affinity process. This is another example, and the last two of them are actually the out of the dataset examples, meaning that these protein ligand complexes have never appeared in the training or test dataset. As you can see, even though it still needs some improvement, it can at least somehow reason which of these residue regions can have an actual NCI with an unseen ligand. This is also another case study, which though the protein regions seem to be scattered across the whole structure, some of them seem to suggest potential binding sites for another ligand as well. However, there are lots of limitations we have thought about after completing this work. First of all, ArkDTA didn't fully excel in the binding affinity prediction task compared to other baseline models. In fact, it didn't outperform state-of-the-art models such as attention DTA. And also, ArkDTA does not take multiple types of non-covalent interactions such as hydrogen bonding and water bridges, etc. into account. And the PDB-bind dataset for primarily training the ArkDTA model suffers a sparsity problem compared to the other datasets such as Davis or METS, which doesn't explicitly provide NCI information. There is a lot of room left to improve the ArkDTA model in terms of model optimization and architecture design. The set transformer framework we have used has its own drawbacks, and multiple objective optimization could be a hassle. So our future work is the following. Identifying potential binary regions prior to identifying non-covalent interactions within the attention mechanism. We also plan to help the model identify multiple types of non-covalent interactions for which chemical substructure to which residue. We also plan to revisit the representation scheme for proteins and ligands while maintaining the structure-free inputs. We also plan to overcome the dataset sparsity issue, which is prevalent in most DTI studies, by using transfer learning or pre-training, etc. Perhaps the ultimate objective of this future work will be improving the model architecture design and devising effective optimization strategies. This is a summary for this presentation. Thank you very much for listening to this presentation. If you have any questions, please feel free to email to the following addresses below. Thank you very much.